start. Thanks for coming, everybody. So this afternoon's seminar is given by Richard Morris, who's a professor over in the John Innes Centre for Plant Science, which is located that direction. About 10 minutes walk over there. So Richard is a long-term friend of the School of Mathematics, has interacted with many people in our department, and is a visiting professor, I think, in the School of Maths. Anyway, so Richard's going to talk about predictive modelling of phase separation.
Thank you very much, Mark. So our understanding of the cell has changed radically over the last several years. So if you open a textbook on biology, you see something like this image. That's a plant cell. And it's nicely organised into these organelles with these different coloured structures here. These are membrane-bound compartments where specialised reactions occur, like photosynthesis or transcription. But recent discoveries have shown that it's not just these membrane-bound organelles that are inside the cell. The cell is constantly forming, making, dissolving liquid droplets within the cell. Liquid-liquid phase separation leads to biocondensates. Within these condensates, you have highly controlled reactions occurring, like translation, transcription, nucleus, example of this. So it's a much more dynamic structure than I think anyone imagined a few years ago. And it's about phase separation.
Yeah, you need some droplets are formed, they make some reaction, and the decollate somehow, there is a little bit depending on the function they want to do.
Correct. So you get hundreds of thousands of proteins, usually with RNA, coming together in the same location. So for instance, when translation happens, you have polymerase going along the RNA, and this will form a condensate, locally increasing the concentration of all the components, will do its job and then dissolve. Respond to temperature, heat, stress, pathogens, all undergo similar mechanisms. So, and that's how I want to tell you a little bit about today. There won't be much maths in this, but there's a little bit of computation. So before I tell you about what we've done in this space, first of all, thanks to the people who are involved in this. This is part of a large ERC consortium, and there's a subgroup within this consisting of myself, you, Leo Kerr, Stefan, Anna, these are experimentalists, and then myself and Ruth doing the computational work. And it's basically all of Ruth's work I'm going to tell you about today. Unfortunately, you couldn't make it here to answer the questions for me, but I'll try my best anyway. So, we're interested generally in transport within plants. So plants have a vascular system, like we have blood, and within that, they transport molecules. And what you can see, maybe see in this picture, this is an image of a plant tissue. There's a slightly, I'm sure you can see it, but there's a slightly different coloured light grey stripe. That's the plant phloem. It's the vascular system that takes photosynthesis from the leaf to the root and transports molecules. They also transport proteins, RNAs, and many signaling molecules. So that's the process we're interested in. And our collaborators recently found that one of the proteins that resides in the phloem is this protein I'm just referring to as parcel. You'll hopefully get what these other words are throughout the talk, but I'll start with the parcel protein. It's a protein that's in much higher concentrations than any other protein in the phloem. We wanted to see, well, what does it do? Is it act in transport? So what our collaborators did was they took this protein and they attached something called GFP, green fluorescent protein. That's basically a fluorescent marker to this protein to see where it is. And it's not uniformly distributed in the phloem. What you see is foci. So this protein seems to sort of lump together in these bright bits here, you can see. And these are several microns apart, so quite large, much bigger than protein. So what are they doing? What is that? Aggregates, what's going on? So that's what we're trying to figure out. And this topic of liquid phase separations is becoming of great interest to lots of people in biology. So the question is is this a liquid phase separation process? That's what we're asking. And indeed, our experimental collaborators then verified that that's the case. So they looked in vivo. So this was the previous picture was in the actual plant. This is looking in the controlled lab conditions, and you see that you get these clusters forming these nice spherical droplets that behave like water, like liquid droplets within a fluid. So the liquid fluid droplet within another fluid with a different concentration of the protein. And these things will slowly merge over time, they will get bigger, they behave exactly like liquid.
Excuse me, can I just ask? So the coloring molecule, the fluorescent molecule, is it smaller than the molecule?
Yes, yeah. It's you'll see a picture later. It's also a protein, protein, but it's quite a bit smaller than the actual protein that we're looking at. But that's a good question that we'll get back to soon, because that's everyone. Oh, I can give you up front. Everyone who's looking into this uses this label. So we were concerned, does the specific label might that actually reduce phase separation? We'll get to that later. But what you're seeing here is the protein with its fluorescent tag, and there'll be thousands of them in one of these. This is there's lots of activity going on in polymer physics and mean field theory, trying to understand this whole process. It's a fairly new phenomenon. So the phenomenon is not dissimilar, there's a difference, but it's not dissimilar from this is balsamic vinegar and olive oil. We're supposed to use the liquids, inking water, gin tonic, whatever, mixing nicely. But there are some examples that sort of stand out as they look a bit freaky, because we're not used to seeing this behaviour. And that's typically these are not stable states. You have to shake the liquid with vinegar to get these droplets. But you do see several liquids that don't mix. They're just not mixable liquids, or unmixable liquids. And this is basically driven by surface tension and the molecular interactions, as you'll see. So we wanted to know which features of this protein drive this phase separation process, and how does biology control that process. If biology is exploiting this process, it would have found ways to manipulate the proteins and the molecules to regulate the phase separation. So that's kind of the question I'd like to sort of through. So this was a little bit basic on the left in anyway, just to sort of set the scene. But what drives this liquid phase separation? So we think about a liquid, quite different from a gas. An ideal gas, no interactions between particles. Liquid have very strong interactions between the particles, changes in motions, the number of neighbours are much more constant. And this influences both motion and the whole, the physical properties of the liquid. And if you mix in two liquids, most of these interactions are about the same-ish. So they will just mix, again, inky water, or whatever, it's milk in tea. Liquids will mix and you'll end up with a fairly uniform, homogeneous distribution. But it could be that some of these interactions are markedly different between particles, in which case what we'll see is that some of these particles tend to stick to each other a little bit more than the others, and then you get these kind of groups forming of particles that tend to co-locate. So at that level, everything is fairly simple and straightforward, it's just about interactions. And what this gives rise to these different phases. You have one phase, so liquid phase, and in that second phase, another phase. And this is where it starts to become a little bit confusing when you think about it at this level, because what this means is that a liquid, most people would model it as an incompressible fluid, but this means that we have, within the same liquid, we will have, area will have two phases of different concentrations of certain molecules, in this case of proteins. Different concentrations can only be maintained with diffusion with different chemical potential. So within the same area, different chemical potential, different concentrations, different pressure. So incompressible fluid, obviously not, because you have different pressures inside and outside. And that gives rise to surface tension. And that's a big interest in people to feel at the moment. How do you get this surface tension emerging? How can you try and compute that? So these are all sort of somewhat maybe non-everyday observations that lots of people seem surprising. Any concentration gradient should, if it was just that, balance out through diffusion, but there's a surface tension that comes into play. But again, just about interactions. So we need to understand the interactions between the particles to understand this process. Once we're talking about interactions, as of course with almost every physical phenomenon I can think of, biology has found a way to exploit that. So biological systems can change these interactions. They can add other bits to the protein, phosphorylation, acetylation, or it can change the pH of the system. So that means that they can regulate and start controlling these processes for their own benefit. And so this is now back to the parcel protein. This is just looking at this, how much you change it. So one thing is if you change the concentration. So this is looking at different concentrations of the parcel protein from very low concentration, 1 micromolar, where there's not much happening, and you start to see you get more and more of these droplets forming a high concentration over there. So consistent with what we expect from the interactions, the higher the concentration, the more you bring them together, the more they stick together. There's a concentration dependence there. But what features of the protein are actually driving this? So there's different approaches that have been used to study phase separation. There's the high level thermodynamics, maximum entropy, I think we touched on. The more sort of polymer physics, medium field theory approach. And then there's sort of down through the sort of molecular dynamics, Newton's law, down to quantum mechanics. Can't be very far with quantum mechanics given the dependencies on electron number, but there's small bits that you can do that. Indeed, small subsections of systems have been used to extract parameters for the next level up. So what we want to do, we understand that a protein is basically a polymer, think of amino acids. Which properties are driving this phase separation? So to put Goldilocks on this is just about the right level. So this is a level that captures phase separation at a level where we can take into account the chemical units of this system, the amino acids. And that's what we want to understand. So we're going to use coarse-grained dynamics to try and understand this phase separation process. What does that mean? So a protein is a polymer consisting of units, 20 amino acids. And some of those are shown here. A long chain typically folds up into a complex globular structure. And if you do molecular dynamics, you take all of the atoms into account when you do the simulations. That becomes far, far too costly computationally to do for, if you did it for one protein, you can do it for two. You can't do it for hundreds of proteins. So what you do is what you can do is you can coarse grain this. You can take groups of atoms and you can approximate their properties by higher order parameters. In this case, largely things like charge, hydrophobicity, and you can attach each of these spheres, which is a different size of every amino acid. You can approximate their properties with these higher level properties. So this is a well-established technique. We're using one of the latest implementations called Genesis, and we've used a recent hydrophobicity scale to model these interactions. Okay, so to start these simulations, you need a structure, so three-dimensional coordinates of protein. There are lots of proteins in the protein data bank where you can just go and say, that's my protein, and start this. This is a highly disordered protein, very, very flexible. There's no X-ray structure of this protein available. We can use AlphaFold to predict it. I'm sure most of you will have heard of that, big in the news. AlphaFold basically changed the structural way that we think about, or we do science with predicting, using AI to predict 3D folds from the sequence. And it does remarkably well for folded proteins, a little bit less so for disordered, but it still gives you a set of coordinates which is fine. So this basically means there is no structure to this, it's just a big floppy chain. These things are called alpha helices, a little bit of structure there, but it gives us a set of coordinates to start with. So we can use that to go from protein sequence to a prediction of 3D structure. And then what we can do is we can make lots and lots of copies of this protein and just put it in a huge big box. So this is what's called a slab simulation. It's a big box full of identical units of different orientations of this protein. It's a slab simulation just to, so it gives you space to explore in this case in the x-axis, but limits the concentration there's a periodic boundary condition on the other axis. So just kind of kick-starts the whole process a bit. So we're starting here from a completely sort of uniform distribution of these units. And then we just run these dynamical simulations for a very long time. So I'm going to just show you snapshots of movies which I didn't in case something went wrong. But we're basically sort of showing you snapshots every 10,000 simulation steps, which are these three of these. Okay, so starting from uniform, this is just this protein, partial protein, bulk subtle taken into account for directly constant. And we see already after 10,000 steps, they are kind of grouped together. Another 10,000 and actually form very nice units. So we're pretty happy with that. We can reproduce, at least at this level, the key features of the system. They are grouping together somehow. So we're pretty happy with that to start with. And then we talked to the experimentalists and said, but that isn't the system that we're looking at. And that's back to your question, so there's a fluorescent tag to it. And no one seems to have simulated this before. So it's not just the protein, it's the protein with this fairly large other protein attached to it that gives us fluorescent marker. So that's caused a bit of a headache because that's a very structured bit of the protein and a very, very long floppy bit. Which you have to have two different force fields, one for the globular bit and one for the very long floppy bit. But Ruth persevered and filled our high-performance computing cluster with many, many simulations and to try and see what happens. So this is the same picture I showed you before of just parcel, but now this protein has this fluorescent marker. And the fluorescent marker is in yellow. So it's a protein with this fluorescent marker. So will that phase separate and form these liquid droplets? So that's good news. So we now have basically an equivalent to the experimental system. You have the protein with the tag and it nicely phase separates. So we have basically a parallel system. Everything we do experimentally, we can do in silico. Every mutation, every change to the physics of the margin make in vitro, we can mimic in silico. Just try both and see what happens. So then the question came up, well, is this type possibly even promoting phase separation? Is it not something biological? Maybe it's an artifact of the system that you have this tagging on. So we did the whole simulations with this fluorescent tag, but not stuck to the protein, just in solution. And if it was promoting phase separation, you'd expect that this tag becomes part of these biocondensators. But that isn't what we see. We see that parcels still nicely phase separate, and these fluorescent tags stay largely in solution. They come in and out occasionally, but they're largely in solution. So that suggests that it's not at least greatly perturbing the system, and it's parcel, this protein that's actually driving phase separation.
You have any idea why they're still in solution and not clumping together?
So these are largely globular proteins, and what we'll get to, it's a great question a bit later, is that what you have to have is something called multivalency to drive phase separation. So this is basically every bit of the protein has to act with many, many other ones, like in a liquid where you have lots of different interactions. Once in a globular state, it's kind of folded up, and all the bits that make these contacts are buried more in the centre of the folded protein, so you have much less multivalency on the outside of the surface, and therefore, given the choice, it's going to be happier in solution. So we fold into a sort of solution-friendly structure. Whereas if you did unfold that, you'd probably go into this state. The fact that it was in the condensate before is simply because it had no other choice. It was attached to something that wanted to form these droplets, and it just to be around.
Sorry, no, so you didn't tell us what are the equations of motion. What are the equations of motion? I mean,
okay, yeah. So it's basically Newton's laws for every normally for every atom in the system. This is for every sphere in the system. And molecular dynamics, you typically have it's Newton's law constrained by bond distance, constrained by angles that we know. We don't have those in here because we're grouping all those things into spheres. So basically, it's a chain. So you have those constraints, and then it's just Newton's law giving every atom a kick in the system and solving those equations.
And for the liquid?
For the liquid, we have a bulk approximation. We just have a dielectric constant, which basically just damps the interaction between the particles. Thanks. Yeah. Okay, so that doesn't seem to be driving the phase separation. So this is basically the picture I showed you before. What you might have seen is that in the simulation, all of these, or most of these, fluorescent tags seem to stick to the outside of the protein. So this was very, very difficult to simulate because you have this globular structure and you have the flexible bit. It's very long time to simulate. It's probably an artifact, right? If you have, it's probably an artifact of the way I was trying to up to Mark's question. There aren't multiphase interactions there. It's stuck there. This is what's driving it, and it's this mismatch of force fields that's driving it. It's still doing the right thing, but it's probably not real. As in, we think this is probably just a simulation artifact. There's lots of approximations to the analysis. Okay? So, but still, does roughly the right thing. We then went back to our experimentalists and said, well, it does the right thing, but can't trust everything, and these things are probably able to take too seriously. And yeah, and they said, well, but we can test that. And I said, well, how can you test that? And what they have is, in biology, you can go through called antibodies, which are basically derived from our immune system, which recognize certain molecules. So they have an antibody which recognizes this fluorescent tag. So if you put an antibody into the system and it binds to the condensate, it must have recognized the fluorescent tag somehow. If it recognizes the fluorescent tag, the tag must be accessible to the surface. So that's the idea behind this. And this shows, this is just YFP. These are these nanobodies, they're about little balls covered with nanobodies. And if it's equally distributed everywhere, you'd see something like this. Whereas if it's binding to condensates, as you see here, you should see what you see here, which are these localized, it comes across. You can basically see these in this ionoxide. It's nice to these different condensates. So what this means is that this antibody must have access, in order to bind it to see it, to the fluorescent tag, which at least is consistent with the tag being on the surface. So maybe this wasn't an artifact after all. Maybe these, which have a structured unit in the protein, kind of tends to be on the surface. So that's actually consistent with that. So at that point, we went through enough kind of controls. We also did this for many proteins that are known not to form condensates. We did the same thing, and they don't form condensates in silica. So then we have negative controls, positive controls. We can reproduce the experimental system, and we made this prediction of GFP present being on the outside that was at least consistent with the experimental results. So we've got at least some trust that maybe this is doing something useful. So that's the point where we can go in and say, well, okay, if this is really doing something useful, which bits of this process, which bits of the protein are driving this process? And the idea here is very simple. If you have a liquid moving around and these tend to interact and join together, it's going to be where they interact that drives this process. So, and we have, through the dynamic simulations, we have gazillions of times frames of particles moving around, interacting with one another. So kind of all we need to do is go through and see where are the most interactions, which bits of the protein are interacting with the other ones, and those are likely to be the ones that are driving phase separation. So that's the idea. So what does this mean? This means taking this big polymer chain, like our protein of interest, and going through every residue. So these amino acids are called residues. Residue number 0, 1. We go through each of them, and throughout all of the simulations, through all of the frames, we say, how close is this to all the other ones? And then we can track those distances and estimate how often this one interacts with another one. So doing this for every residue, 1, 2, through the whole chain, with all other chains in the simulation, through all time steps in the simulations. And then what you can do is you can produce a map like this. So this is the number of amino acids in the protein, and this is the mean count that it interacts with another protein. So if there was any information in there, this wouldn't be flat. Flat just means they all have equal contribution. So let's look at what we get. We get something like this, which is already quite interesting. So first of all, it's not uniform, not flat, that's good. Might be information in here. There are these regular peaks here. So this isn't quite nice. So this fits with one of the main models of polymer physics called the sticker and spacer model, where you have to have, in your polymer chain, you have to have bits that can technically do nothing, and then you have to have sticky bits which combine to other sticky bits in the protein. So at least at this high level, we're kind of seeing a sticker spacer model in there, which is kind of nice. Before we go on to over-interpret that, another check that we can do is note this protein parcel binds to another molecule, it's a microRNA, and we know where it binds. So we can throw this microRNA into the system, we can do all the simulations that phase separate some bits in, some bits out, and then we can use this to verify whether this interaction idea makes any sense or not. So we can look at the same thing, which residues interact now with the microRNA, and we get these hot spots right at this end here that seem to be indicative of where these interactions are driving phase separation. That turns out to be exactly where the microRNA binding site is, suggesting again that there's some use in this idea. So with that, we then went on to look at this in more detail, and it turns out that all of these peaks are amino acids called tyrosines. They're a big, bulky amino acid with a flat ring, and these are known to form pi-pi interactions, which can drive phase separation. So again, it's looking pretty plausible. This is telling us something. It's picking out the sticker spacer model. It's picking out the right types of residues. Furthermore, this whole area with this enhanced interaction map is in something called the prion-like domain, which are known to form aggregates and phase separation. So again, it's all looking pretty consistent. So, based on that, what we can do is we can take the protein and say, well, let's take all of these key amino acids out, or change them to something else in the simulation, see what happens. If we change them, if they were important residues as they were, and we took them out, then something should happen. So Ruth, in silico, mutated all of these amino acids in the sequence, and reran all the simulations again, and nicely, as she predicted, is that no longer phase separate, suggesting again that these amino acids are important for this phase separation process. And even nicer, it wasn't just confirmed in silico, this is an in vitro confirmation of parcel, that is the wild type, it's called just the standard protein. And over here is this mutation that Ruth introduced. And there is a little bit, I can see it very nicely on the screen, but there's not half as much as to that. And what you see in the middle is also quite interesting. So this is a phosphorylated protein. So I said that biology has learned to change proteins to regulate their phase separation behavior. Parcel can be phosphorylated, basically it sticks a phosphate on some of the residues. If you stick phosphates on the residues, that becomes, in terms of the charge and mechanical properties, exactly what Ruth did here with the mutation, and you get the same physics and you get the same behavior. So this is a way that biology can then just change the protein and then change the whole phase separation, which is kind of neat as well. So experimental confirmation of what we thought. So pretty happy with that. So that's several several things of that model look like they're okay. Stick has spacer, tyrosine picked out, mutated tyrosines gives different behavior and verified in vivo. What Ruth did then was to say, well, if that works, why don't I just try all possible combinations of mutations? So this was, I don't know how many weeks of work on the HPC, probably months. But anyway, she went through all possible combinations of these residues. And for each of the residues, quantified kind of how well they face them. So this is the one that I showed you previously. But you can get all these different types of these different levels of how you can change this droplet formation based on these different point mutations. And I think we've got 3, so this is still a bit outdated, we've got 3 of those confirmed in the meantime. The numbers are kind of meaningless, the numbers we can't measure, but in terms of how much they phase separate, the number of droplets and their size, we can give a ranking to them, and it's the ranking that we're looking at to be consistent with their predictions. So if you want to read more about that, we've done that on the bioarchive. We just go through the whole process I told you about in silico simulations and validations. So what have I told you? I've told you that this process of liquid phase separation is very common in the cell. There's every process that's been looked into, almost every process, people find these biocondensates, find liquid phase separation as a means of responding to temperature, to different stresses, and locally controlling reaction rates and concentrations to optimize whichever process involved. And yes, if we did a modern drawing of the cell, we'd be seeing phase separation and biocondensates pretty much everywhere. It's really amazing how much the process is involved in. And it's a key area is trying to understand what drives that. And the way that this was done previously, people just tried random mutations. So what we think we have now is actually a very targeted way of using these simulations to understand which residues are driving this process and actually go and doing all the sort of the biology in silico first, see what it does, and then verify it later. So it's a very nice approach for engineering proteins that are designed to do what you want based on these simulations. So what have we done in terms of what we've shown? So we've shown that YFP, this replicating by itself, doesn't phase separate, that this protein that we've found forms condensates. We can reproduce these condensates in silico, and we also showed that with the YFP tag, it also very nicely phase separates, possibly with this tag on the surface, that's at least consistent with the other data that we have. And importantly, I think, with the outcomes of this is that we'll put this into a package which allows you to go from sequence to predicted structure to doing all of these simulations in a fairly automated way, and then you go through the contact map analysis and looking for interactions. That highlights key residues. Doesn't tell you how to change them, so you have to think about then a bit about the biology of which residues to change to what. But that will give you predictions that you can then again go through the same pipeline, simulate, doesn't do the right thing. If it does the right thing in silico, then that's the time to go and see if that's the case. So this gives you a neat way of picking out key interactions, key residues, and a way to in a targeted manner to engineer the protein of your choice to phase separate. I should point out this took a huge amount of time. So what Ruth is interested now in, so her background is protein structure, but also machine learning. So what she's trying to do is, and this is looking quite promising, she's using latest autoencoder techniques to try and go directly from sequence to predicting this interaction map without any of the stuff that takes such a long time. So it's a long shot, but if it works, this would be pretty neat. This would be blazingly fast. Sequence, contact map, predictions, phase separation. So that's what she currently after all that effort, we still don't actually know what this protein does, which was the way we started off. So there's still lots of work to do on this. What I didn't tell you, when I showed the first image, was that this protein moves in the flow up to about 100 microns per second. So it's possibly involved in the transport of other molecules and or transport of RNA. And we're looking into that now and seeing how we can use what we're doing here to perturb the system and understand how it affects the phenotype. But we know that particle forms these biocompensates, we know that it binds RNA, we know that it moves, but what it really does, we don't know yet. So that's still ongoing. Thank again the people involved with this. It's a really fruitful collaboration. I think this interaction of the sort of simulations with experiments has worked really well. It's been key for trying to understand the system. And then I'll just thank all the generous people who funded the work in my group. And with that, I wish Ruth was here to answer the questions for me, but I will do my best.
Any more questions?
Presently we have a liquid phase separation, where you have a droplet of one liquid that can grow. There is the dynamics of coalescence that can have different mechanisms, reservoir weakening, or did you identify kind of such a dynamics, or is it just particles that somehow
yeah, really good question. So the Oswald
adhesion or coagulation?
Yeah, I don't know. So great question. So the Oswald brightening you can see in the vitro experiments, and if you run the simulations for longer and longer and longer, you start to see the number of droplets gets fewer. I don't know why. I don't know, but I presume it's the same types of interactions that are driving it. But I can it's a good question.
Similarly, you mentioned that there is a surface tension in the system.
Yeah.
We can get that if we do, let's say, a phase field model, which is not what you're doing, if I understand. So do you get idea that at some point of the surface tension that you can define your system based on?
So we don't get the surface tension from this. There are ways of looking at the density of molecules and kind of getting an estimate of what the different concentrations and this is getting very, very hard-wavy now. Pressures might be, but that's a very hard area at the moment. It's trying to go from the physics of the polymers to predicting basic surface tension. So that's a big brain in that. So I'm hoping someone might want to do that.
When a phase field modeling, the surface tension can be understood as the energy to go from one state to the other one. So there is a cost to build this interface. This is an energy per unit line in the surface, and that's the surface. You may have that, or is it maybe an entropy in your case? I'm not sure. It might be something to be found at the end.
Yes. Yeah. Again, I don't know. So this, that would be a great interest if we can try and get that. We'd have thought that you're right. If you get where you do, if we looked at one droplet and one separated unit and did lots of simulations over the time of that, we should be able to compute the difference in free energy of the system in the isolated state versus in the droplet state, and that will at least give us an estimate. We haven't done that, but that's a good suggestion.
So in your simulation, when do you know that it's safe to stop?
Yes, we don't. Yes.
So,
yeah, we don't. So we run it for something like 1 million time steps. That's what the bottom was. I can't exclude that if you run the other ones for even longer, it might. And it also depends on the so in principle, if you up the concentration and change the temperature, everything should somehow interact with all these things. So there's a lot more to explore in terms of the external parameters. This is all for one temperature, so my statement about this doesn't phase separate. It should have been a bit cleaner, is that under these conditions, under this temperature, this concentration, it doesn't phase separate, and under these time scales, it doesn't phase separate. So we just run it for as long as we're kind of willing to wait to see what happens. These first droplets seem to come fairly swiftly, so this is going to go something like 500, it's about 10,000 steps. You do something, so that's the tendency is there, and the other ones.
I was more concerned about the fact that these particles they could continue to grow. I mean,
Correct, yes, yeah, and they do, yeah, so,
right.
so running it for even longer reduces the number of, so in the one which is just past that happens, and the one with GFP it doesn't, so the GFP seems, so that's the first attack, seems to actually, if anything, slightly inhibit the phase separation, or add or slow down. So you have to run it for even longer than to get to that stage. But you're right, yeah, and this is Drew's question about slow, right, because you might get, or even longer, you might get those.
Okay, that was a very interesting, very nice presentation. I'm not an expert in that field, but I know from chemical engineering there's a big, well, for the past 10, 20 years ago, there's a big tool about bridging the gap between molecular simulations and predictive macroscopic properties. So since he raised, our colleague raised the question as to where you stop, isn't there some macroscopic property of the protein or the plant that you're looking for that you're eventually trying to predict, so you can kind of start with your molecular simulation, do some coarse grading, and eventually come up with the holy grail, which is prediction of macroscopic properties. I don't know if that's possible.
Yeah, no, so it's still a really big question, bridging that gap, and that happens both that gap is both at time scales and at spatial scales. And what we have here is a kind of compromise situation, so that we'd be interested to predict the given concentration properties, the number of these droplets and their size. We can't, currently this isn't accurately captured by the model. It's
Don't you use coarse grading procedure through which you can gradually move up, no?
So yes, we haven't tried that. That's a really good. So you could envision that you do this to a certain level and then learn from that what the rules would be to approximate at the next level. A kind of thermodynamic approach to working up.
I've never done it. I've just heard other people doing it in polymer
Yeah.
science. I was wondering if it's possible.
I don't know if anyone's done it in this area, but
Okay.
in principle, the idea and the concept, absolutely, and that you could go through a series of course grades at different levels and get to something like that. And if that was then compatible with meat field theory, for instance, that would be pretty cool.
So, Richard, if I understand correctly, the suspension fluid in the background, which is water, that it's floating.
Yeah.
That's static, effectively.
Yeah, so
But
that's
actually the flow is moving.
that's
There is some sort of sharing flow, would it make a
I
difference?
meant to mention at the end of this, I forgot. Thank you so much. Yes. So in all the simulations, it's static fluid. In the flow, it's of course not. It's this sticky sugary mess, which is flowing at 100 microns per second. So what that flow effect is on the condensate would be really interesting, as in, do you get these boundary conditions where you kind of maybe that flow and you're actually kind of driving this formation of the droplet, and why are they even in these droplets? Is that more stable flow? Does it label fill all these? I don't know. So that would be, in terms of the function, that's a really interesting question. How would you account for that in these simulations? Maybe that leads back to the sort of more coarse grain we have to go through. How would you combine a moving fluid with all of the key properties of that sticky, sugary, viscous mess with these condensate formations? And I mean, are they even, they have to be robust because we can see them, but in the model, I don't know if what would happen if you had a asymmetric force applied to these condensating molecular dynamics. I can imagine being ripped apart. That's a great question, but I don't know maybe that would be very interesting.
Okay, should we thank Richard again?